been happening before me. And so I'm really honored to be part of this group and to give you sort of our view on the type of brain prediction, some of the brain prediction studies that I think will be really exciting moving forward. And so, of course, a core question that I think has brought many of us here today is to try to understand how different aspects of brain structure and function underlie cognition. And we can investigate this sort of question in a number of different ways. And I'll sort of just illustrate two, what I'll say as opposing paths, of course, that maybe is a simplistic view. That is, on one extreme, we might try to investigate how, we might try to answer the question of, say, what does brain network X or brain region X do by looking at population level variation in that network and relating it to population level variation in different behavioral features. And as many might be aware, recent studies have suggested that we maybe need very large sample sizes in order to do that sort of study robustly. And instead, I'm going to focus a little bit on today a different set of approaches that I think have been really fruitful throughout the history of cognitive neuroscience, where we try to address these types of questions, often by looking within subject at how variation in the activity, say, of a particular brain region or network over time may also relate to different types of cognitive or behavioral phenotypes. And so I'm particularly going to focus on one extreme version of these types of studies that I'm going to call precision fMRI, and give you sort of a whirlwind tour of why I think precision fMRI is cool and what it can do to help us sort of improve brain prediction studies in the future. And so what do I mean by precision fMRI? Well, in any given study that we design, due to practical and budgetary limitations, we often can only collect a certain number of MRI hours. And traditional studies have often focused on, say, collecting 1 hour of data from 20 to 30 participants in fMRI. And of course, we've heard some examples of really great studies that are pushing the bounds on how many subjects can be collected in single fMRI experiments, doing these large population level studies, but usually at the cost of having relatively little fMRI data per person. Now, in precision fMRI studies go the other direction. So rather than collecting a very large amount of data, or sorry, a smaller amount of data from a very large sample of participants, they collect a very large amount of data, usually on a relatively small number of participants. And so this has sometimes been called deep fMRI or precision fMRI. There are many terms. It mostly refers to the extensive sampling of these individual participants. And it's often accompanied not only by extensive sampling and fMRI data, but also phenotypic and other types of measures. And I'm not going to go into any detail on this slide because I think that we have been talking about it quite a bit over the course of today and also yesterday. Besides to say that I'm going to focus on applications of precision fMRI, particularly in the context of measuring functional brain networks from fMRI. And I'm happy to come back here in the question session if anyone has any comments on that. And I'm in particular going to focus on two data sets that we have found quite useful in the past. One is the myconnectome dataset that was collected by Russ Poldrek on himself. He was scanned twice a week over the course of about a year, with accumulating quite a bit of resting state fMRI data, as well as a fair amount of additional phenotypic data outside of the scanner. And another data set that I have been involved in is called the Midnight Scan Club data set. It was collected at Washington University when I was there, spearheaded by Nico Dosenbach and Steve Nelson and colleagues. And this contains data from 10 participants who were each scanned at midnight for 10 times. They were scanned at midnight because the scanner was discounted 90%, making it a very economical study for a young assistant professor to gather. And in addition to collecting a set of resting state fMRI scans from each participant on each day, they also completed 4 other task studies. And so you might ask, why do this? Why have this enterprise where you collect quite a lot of data from single participants? And I'm going to highlight a range of results from different groups that I think illustrate the power of these approaches. So one real potential from precision fMRI is I think it improves our measurement of brain networks. One obvious way that has been, I think, discussed quite a bit in the literature is its ability to highlight the level of reliability that we can achieve with different functional MRI measurements. For example, Tim Laumann very nicely illustrated with Russ Poldrack's MyConnectome data set, that with this large amount of data, you can produce this very nice and detailed map of his individual connectome that achieves very high reliability if you were to split the sessions in half over the course of the year. And indeed, you can then use these sort of measurements to sort of back estimate how much data do you actually need to get a reasonable level of reliability for that connectome. And so this is a curve that Tim has created, for example, estimating the level of reliability that you have with X amount of minutes of data of fMRI. And of course, this can be extended beyond just asking questions about this functional connectome matrix as a whole, to also looking at the reliability of many other types of metrics, some examples of which are shown here from the Midnight Scan Club dataset. Now, these approaches can also be used to define individual specific brain regions and networks, which I would argue can in turn increase the validity of our brain network approaches, and in particular, increase the validity in the sense of identifying separable functional anatomical units. So for example, when we define individual specific motor networks using these sort of approaches from precision fMRI, you find that they have very good correspondence to activations during motor tasks, and much better correspondence within individuals to their own motor task activations than that seen across individuals or to the group average. This has also been shown to extend to higher level task contrast. I'll just note that these sort of approaches are likely to be important to understanding functions of higher level regions of the brain, as discussed by my former postdoc, Derek Smith, in a recent review article in the context of the dorsal anterior cingulate, which is perhaps one of the most heavily argued over regions of the brain. And we see within single individuals that we can find many closely juxtaposed networks that differ slightly in position and shape across individuals. So we don't account for this individual variability. We risk mixing signals across these different people. So jointly, I'd argue that this enhanced reliability and enhanced validity of our measures should lead to better brain prediction, better behavioral prediction that we can expect from fMRI. And so these approaches can then be used to answer questions about brain behavior relationships at the single subject level. Now, given the timing of my talk today, I'm not going to discuss any of these in detail besides to plop a few on the screen. Hopefully, again, we can talk about these later if people are interested. But these sort of approaches have been used to show finer scale dissociation in the functions of different brain networks. They can be used to track how brain organization responds to injury, in this case, to perinatal stroke. And they can also be used to track at a very fine time scale how functional connectivity responds to different forms of interventions, in this case, casting of the dominant arm. What I'd like to highlight specifically, given the topic of the session today, is the importance of these approaches to classic brain prediction studies by telling you a little bit about a project a graduate student in my lab, Alexis Porter, recently conducted, looking at how we can predict task state from functional connectivity measures. And of course, a number of groups have shown that we can do this effectively with functional connectivity. But Alexis was interested in asking, to what extent are these individual differences important in our classification of task state? And so she took that Midnight Scan Club data that you may remember had 10 sessions within it. And she took 9 out of the 10 sessions and used that to train a classifier to predict among a number of different states that the participants experience inside the scanner that are shown here. And she then tested the performance of this classifier on classifying those states with independent data from that same participant from a different session, or with data from a new participant, as shown here. And so now when we look at how the classifier performed, we find a number of different effects. So first off, these individualized classifiers that were trained on a single participant were able to successfully classify task state, both within that person as well as different people, showing that there are these sort of generalizable components. However, as may be notable, just looking at this slide immediately, the performance of these classifiers is significantly better on that same person for independent data from that same person relative to other people, suggesting that there are these large person-specific components to classification. And she dug deeply into why this might be, and sort of, I'm happy to talk more in detail about this, but just to reflect that across a number of different analyses, we show that these classifiers are revealing different forms of features than we see in more classic leave one subject out group classification. We find different levels of brain areas and specific connections that tend to be highlighted. And of course, as more public data sets sampling a wide variety of states and inputs come available, they'll be useful for powering prediction studies at the individual level. And I just want to highlight a couple efforts in this domain from other groups that I think might be quite valuable to folks interested in this. One is the individual brain charting data set, which contains data from 12 individuals who were scanned across 150 fMRI contrasts. And the other is the natural scenes data set, which includes data from 8 individuals who were each scanned for 40 hours each while they viewed 73,000 natural scenes. So if this is of interest to you, if this type of approach is interesting, I'd like to highlight a recent special issue that Rodrigo Braga and I co-edited, where there are really excellent contributions from a number of different authors highlighting the power and the potential of these individual focused analyses. And hopefully this might also suggest to you the value of using these sort of precision fMRI approaches in your own future studies of brain prediction. And I'll just close here by thanking my collaborators in my lab for all their work on these projects. I've sort of highlighted here in bold those that I talked about specifically in this talk. And I'll also put my contact information up on the slide in case any of you have questions that you don't want to ask in front of the full group or just are tired right now and something occurs to you later, and you can feel free to email me. And I'll also highlight that my lab is in the process actually of moving to Florida State, and we are recruiting. So if you are interested or know anyone who might be interested in doing this sort of work, do please feel free to reach out to me. Thank you very much.